I hope the movement of substances in and out of the cell is clear to you. Now we need to do the next thing, that we know the cell performs its function, but the cell has to divide. Why the cell needs to divide? For reproduction, growth, and repair. Now, how does the cell cycle takes place? This cell cycle is divided into 3 stages. First is the preparation phase, which is the interphase stage. It is the longest phase of the cell cycle, in which the cell grows in size and prepares the cell by making all the proteins and the enzyme needed for cell division. And the very important step that takes place in the interphase stage is a replication of DNA, where the DNA duplicates its content. After this preparation or the interphase stage is done, then comes the mitosis stage. Mitosis is the division of the nucleus, in which the parent cell splits into 2 daughter nuclei, which contains exactly the same number of chromosomes as the parent cell, so the daughter cells are genetically identical. And next is cytokinesis. It is the division of the cytoplasm, which takes place after the division of the nucleus. Now, what is mitosis? It is a type of cell division in which a parent nuclei divide to form 2 daughter nuclei with exactly the same number of chromosomes as that of the parent nuclei. The daughter cell produced are genetically identical to the parent, and we can call them clones. This division is important for growth, regeneration, and repair, and mitosis is also important in asexual reproduction. Now, how the stages of mitosis takes place, here's a diagrammatic representation of it. We started with, although there are 46 chromosomes, 23 pairs, but for diagram, we are starting with 2 chromosomes. So these 2 chromosomes first duplicated. Once they duplicated, they lies at the center through these spindle fibers. The spindle fiber shrinks and breaks them from the middle, so that each end now has 2 chromosomes copy, and then the nucleus divides. So we get 2 daughter nuclei with exactly the same number of chromosomes as the parent nuclei. Okay, so I hope this mitosis is clear to you. You should know what are the stages of the cell cycle, how the cell divides, what is the importance of mitosis, and what is the significance of mitosis. Okay, now let us see how the development of a human cell takes place. Now let us look at how the process of cell specialization takes place. Now, let me first explain you what is the entire life cycle of a living organism. Now, we have a male parent and a female parent. Now, both male parent and female parents have 23 pairs of chromosomes. This is what you need to remember. We have 46 chromosomes, 23 pairs, from which 23 has come from our mother and 23 from our father. Now, male cell, they undergo another division. We just saw mitosis. There's another division called meiosis, which produces half the number of chromosomes and is required to make the gametes. So the male parent makes a sperm or a male gamete, and a female parent makes ovum or a female gamete, and both has 23 chromosomes. Now, during fertilization, they both fuse to form a zygote, and this zygote now has 46 chromosomes, so the 46 or 23 pairs numbers is restored. Now the zygote divides into 2, 4, 6, 8 and many cell types and forms an embryo. Now at this stage, the entire cell is unspecialized. It has no function. We do not know which part of the cell will form the digestive system, which part of the cell will form the circulatory system. After that, the process of cell differentiation begins. Now this embryo, which cells have not yet specialized, are stem cells. So stem cells are the undifferentiated mass of cell which can differentiate into any cell type. Now after this, what happens is the process of cell specialization, where the cell becomes specialized to perform specific function. So some part of the cell will get specialized to form the circulatory system, some will specialize to form digestive system, and will give rise to a whole new organism. So in plant cell, the majority of the plant cell are the stores of stem cells. The root meristem, shoot meristem are the parts of actively growing part of the cell which contains the stem cells. We can take that and make a new plant. The plant can be grown easily because majority of the parts are not differentiated, and the ones which are differentiated, their differentiation is not permanent. So they can be reversed. Okay. Now in animals, majority of the cells are differentiated at a very early stage. In a plant, they get differentiated at a very late stage. So as in plants, they are differentiated at a very early stage. they are already specialized to form a nerve cell or a muscle cell. Now, adult cells replace the old and worn out cells in humans, but they also have a limited regeneration and the specialized power. And as majority of the differentiation is permanent, the cloning of the animals is difficult. So this is what happens in an animal differentiation, and this is what are the plant differentiation. Okay, so now, what are stem cells? So stem cells are the undifferentiated massive cells that can differentiate into any cell type. The sources of the stem cells are embryo, leftover remains of the embryo, umbilical cord. These are the sources of embryonic stem cells, and bone marrow is the source of adult stem cells. Now, what is the possible promises of the stem cells? There are a lot of problems in which the transplantation gets failed due to the rejection of the recipient from the donor, because the cell recognizes it as a foreign and the immune system tries to kill it. But if the stem cells of the person body are used to generate the organ, then the chances of rejection can be solved. Second, there are a lot of neurodegenerative disorder in which the part of the nerve cells get damaged. If the person's stem cells can recreate those nerve cells, and the neurodegenerative disease can be treated. The disease like blindness can be treated. It can be a cure of diabetes. If the beta cells of the pancreas get regenerated, it can help in a therapeutic cloning. In a therapeutic cloning, we take the adult stem cells, make it to make the embryonic stem cells, and then embryonic stem cells are specialized to make the desired organ. And it can also solve the organ damage problem. So stem cells is a very good promise, very good potential medicine revolution for future. However, there are a lot of issues against the stem cell. As the cells of the stem cells are rapidly dividing, we introduce the stem cell into an organism, it can lead to cancer. The stem cells can be contaminated and can cause unwanted diseases to the patient. The research in stem cell is still slow and expensive. The research happens on aborted embryos, which is considered as a potential source of life, and many religions have ethical concerns against it. And the knowledge of cell specialization, what genes get switched on and off, on differentiation is still incomplete. Okay, so I hope we have covered this topic in a much detail, and I have tried to cover the maximum of your specification. Now it's the time to test yourself by seeing if you know these key terms. So what are cells? Mitochondria, nucleus, cytoplasm, ribosome, prokaryotic, eukaryotic cell, cell wall, cell membrane, vacuole, microscopes, resolution, magnification, xylem, phloem, diffusion, osmosis, plasmolysis, turgid, flaccid, mitosis, differentiation, stem cell, therapeutic cloning. If you are still not clear with these terms, I would recommend you to go back, watch the videos, and then try over these key terms. So now you can pause the videos and have a go on trying these key terms. Okay, so I hope now you have written these answers on what is the meaning of these key terms. Now let us look at your answers. What are cells? Cell is a basic structural and functional unit of the living organism. Mitochondria is the organelle, which is the site of aerobic respiration. Nucleus is the organelle which controls the activities of the cell. Cytoplasm is a jelly-like fluid which fills the cell and contain enzymes for chemical reaction. Ribosomes are the cell organelles for the site of protein synthesis. Prokaryotic cells are the primitive cell without nucleus or membrane-bound organelles. Eukaryotic cells are the advanced cell type with nucleus and membrane-bound organelles. Cell walls is the outer layer of the plant cell which provides shape and support to the plant. Cell membrane is the outer layer that controls what goes in and out of the cell. Vacuole is an organ present in the plant cell which has cell sap and makes the cell turgid. Microscope is a device that is used to see the objects which are not visible by the naked eye. Resolution is the ability to distinguish between two closely placed objects. Magnification is the ability to enlarge an object. Xylem is a transporting tissue that transports water and mineral. Phloem transport food. Diffusion, movement of substances from higher concentration to lower concentration. Osmosis is the movement of water molecule from a region of high concentration of water to low concentration across a semi-permeable membrane. Plasmolysis is the shrinking of plant cell when placed in a hypertonic solution. Turgid is a fully swollen cell which has gained water by osmosis. Flaccid is a soft cell due to no net movement of water. Mitosis is cell division that produces identical daughter cell. Differentiation is cell specialization. Stem cell is an undifferentiated massive cell that's specialized to any cell type. And therapeutic cloning is using adult cell to produce embryonic stem cells and differentiating them to produce a required cell type. So I hope now you are well clear with this topic. Now what you need to do next is check the specification, make sure the specification is clear to you, and then you need to do the exam question on this topic. If there's still anything that you do not understand, leave a comment below and we'll reply you as soon as possible. Or you can come to our website where we have set up a 24/7 chat support till your exam. You can come anytime, ask any questions, and you can get instant reply. If you like this video, then do subscribe to our channel and do not forget to like, comment, and share this video. If there's any specific topic you want me to make the next video on, then also do leave a comment below and I'll make sure I put that up before your exam. Okay, so I'll see you next in the next video of the next section. Till then, happy revising.